today's webinar, we will be focusing on the products from Ibidi for cell-based assays in cancer research. So during the webinar, I will turn off my video so that my slides can run smoothly. Give me one moment. Okay. Right. A bit overview of Atlantis Bioscience. Atlantis Bioscience is established in 2017. It is a private company distributing life science products and lab equipment cater to the various needs of biomedical and healthcare research. I think, did I turn off my video? No, okay. Yeah, there you go. Okay. So we provide solutions such as cell culture media, animal and human serum, cell therapy, exosome research, flow antibodies, cellular stains from various partners all around the world. So today we will be focusing on the solutions provided by Ibidi for cell-based cancer research. Ibidi is a leader for functional cell-based assays and advanced products for cellular microscopy. It is founded in 2001 in Germany. Ibidi is present in more than 40 countries worldwide. It has more than 450 products and supports over 40,000 customers. Importantly, Ibidi products are reflected in over 18,000 peer-reviewed publications. So what is cancer? I think I accidentally clicked. Okay. So what is cancer? Cancer occurs after a single cell in a tissue is genetically damaged, resulting in uncontrolled proliferation. This leads to tumor growth and subsequent tumor vascularization. Some cancer cells can also become malignant and they acquire the ability to penetrate the walls of the blood vessels and eventually brought to distant parts of the body, forming new tumors. There are various assays to analyze the individual steps of cancer and metastasis in vitro experiments. For example, in immuno-fluorescence assays, they are used to visualize specific biomolecules with antibodies in fixed cells. Movement of cells can also be measured using migration assays for the analysis of undirected cell movement, or in chemotaxis assays for the analysis of directed cell movement towards the source. Blood vessels formation can also be measured using angiogenesis assays. Cells can also be cultivated under flow for the study of rolling and adhesion of cancer cells to endothelial cells. Microscopy is one of the most common techniques used for cell-based assay. One outstanding characteristic of Ibidi Labware is the thin cover slip bottom, which has excellent features for high-end microscopy applications. So in order for all of these assays to work optimally, it is important to use the appropriate or suitable labware for the different types of assays. So Ibidi offers a wide range of labware in different geometries, ranging from channel slides for flow assays, slides and dishes for everyday cell culture and high-resolution imaging, multi-well plates for high-throughput assays, and specialized labwares for functional cell-based assays. So there are Ibidi offers two types of bottom cover slip. One is the glass cover slip, and the other one is the polymer cover slip. Both provide excellent optical quality. So for the glass cover slips, they are made of the Scott high precision glass with a thickness range of about 170 micrometer. These glass cover slips are recommended for techniques such as TURF or super resolution microscopy. The glass cover slips are traditionally used for microscopy, but not all cells attach well, so you might need to coat the cover slips in order for the cells to attach. So for most applications, the polymer cover slip is the material of choice. Its thin plastic cover slips forms the bottom of most micro slides, micro dishes, and micro plates. It has a cover slip thickness of about 180 micrometer, and it meets all optical requirements for microscopes and is suitable for various imaging techniques such as phase contrast, DIC, wide field fluorescence, confocal microscopy, FRET, etc. The polymer cover slip is gas permeable and allows carbon dioxide or oxygen to exchange during cell culture. It also offers improved cell attachment and provides optimal growth conditions for various cell-based assays and cell types. The polymer cover slip is available with different treatments or coatings. Ibidi treat is the most recommended surface modification because it allows most adherent cells to grow well on it without the need for any additional coating. Selected Ibidi slides are coated on collagen or polyl lysine for adherent cells that require specific extracellular matrix. The uncoated hydrophobic surface is not treated and does not permit direct cell growth without an additional coating. Therefore, it is not suitable for the standard cultivation of adherent cells. Due to its hydrophobicity, it can be used for specific coatings or for non-adherent suspension of cells. However, if you would like to culture cells in suspension, we would recommend you to use the bio-inert surface. In contrast to standard ultra-low attachment coatings offered by other competitors, bio-inert is completely non-adherent and does not allow binding of any biomolecule, even in long-term experiments. Therefore, they are good for the culture of suspension cells and spheroids. Bio-inert surfaces are also used in micropatterning, which I will share a bit more in the later part of this presentation. Immunofluorescence is a powerful approach for getting insights into cellular structures and processes using microscopy. It allows us to assess the protein expression and location, making it indispensable for researchers. So in the traditional method, which we are all quite familiar with, the immunofluorescence staining protocol takes a long time and it requires the use of loose and fragile cover slips, glass cover slips. This usually involves the sterilizing of the cover slips, placing them into the multi-well plate, washing, cultivating the cells on the cover slip, fixing, staining, and then after that, you have to remove the cover slip and mount them onto a glass slide. Seal them with a nail polish. So this whole procedure is long and tedious, and it is not uncommon for you to drop the cover slip and break it during the process. And sometimes it just happens to be the most important sample, and you may need to repeat the whole experiment again. So, to save you the pain, we have a much simpler workflow. This immunofluorescence staining protocol that we are looking at now is much shorter than the traditional way. There is no need to grow the cells on loose cover slips. So what you just need to do is to cultivate, fix, stain, and image them directly on the slide. So this has shortened the whole procedure and made things a lot easier. We have several solutions to meet your immunofluorescence needs. One of the most popular products is the chambered cover slips. The chambered cover slips comes in 2 well, 4 well, 8 well, and even up to 18 wells. The chamber slides are versatile and can be used for different cell culture applications. The most cost-effective is the 18-well chamber slide, which only requires small cell numbers and low reagent volumes. You could perform parallel assays of different cell types or conditions without the fear of cross-contamination. Using a single immunofluorescence protocol, you can stain all 18 samples at one go. This will allow reproducibility and consistency for your experiments. We also have channel slides that are especially suitable for immunofluorescence staining of limited samples. The geometry of the channel slides is ideal for the exact exchange of small medium amounts. It provides ideal geometry for homogeneous cell and antibody distribution. So I would also like to highlight that these slides are also optimal for phase contrast microscopy, as condensation inside the optical pathway is not possible. So as you can see from this diagram, so this is the standard well. So you normally would face some condensation on the lid. So with that, but if you look at phase contrast, it won't give you a nice optical image. So another reason for it being good for phase contrast is that there is no meniscus formation using this slide as opposed to the standard well, as seen here. We also offer chamber slides, which are ideal for long-term storage of samples that requires to be mounted with glass cover slips. So this is the glass slide. So eventually you will need to put a glass cover slips on top of it. So this silicone cascades can be removed. So apart from these slides, the micro dishes and microplates are also ideally suitable for immunofluorescence staining and high resolution microscopy due to their thin cover slip bottom. Cell migration is an important step for cancer metastasis. Migration assays are widely used for analyzing cell migration under different conditions. So one of the most characteristic readout of a migration assay is a change of the cell covered area, which is the gap closure over time as seen from this movie. So you measure this closure over time. So to create a gap, we can make use of the Ibidi culture insert. This is a silicone culture insert. It is biocompatible and can be stick to the surface easily. It prevents any cell growth beneath the walls and create a defined gap of 500 micrometer. It is available in different configurations such as 2-well, 3-well, and 4-wells. Unlike other gap creation techniques, the inserts are suitable for co-cultivation assays using multiple cell types or treatments and allow up to 4 technical replicates at the same time. So these inserts are available as individual inserts in a micro dish 25 mm as shown here, or as a 25 pieces in a transport dish for your self-insertion into any labware of your choice. The culture insert 2-well is also available in a 24-well plate for a high throughput assay. So it is really easy to create a gap using this insert. So you just simply stick the insert in an appropriate vessel, in this case as shown here. You then sit and culture the cells until they form an optically confluent monolayer. After that, you simply remove the insert and this will create a really nice gap as shown here. So the cell migration can now be monitored by using live cell imaging or by taking photos at different time points to analyze the gap closure. There are different approaches to create a gap for migration assays. One of the most common methods is to use the scratch assay, where the cell monolayer is manually scratched with a pipette tip or a needle to generate a gap. However, there are several drawbacks with the scratch assay. The gap creation in the scratch assay varies, and it is highly dependent on the pressure applied to the pipette tip and on its size. Scratching removes the surface coating and also damages the vessel. This will result in changes in cell adherence and migration in this area. Scratching also sometimes results in cell damage and may cause unwanted cell death. So as you can see here, the insert provides a more relatively consistent data, while that of a pipette tip, right, is actually rather noisy. The graph is all over the place. Here is one application example of migration assay, which was recently published in Cancer Letters early this year. So in this study, the investigators are testing the effects of exosomes isolated from lung cancer cells on endothelial cells migration. So HUVEC cells, they were seeded in this 2-well culture insert. After incubation for 24 hours, each insert was removed and then incubated in endothelial growth medium containing exosomes isolated from the non-small cell lung cancer, which is these two, A549 and then H460. This is a lung cell, is a control cell line to use in the experiment. So they further incubated the cells with the media containing exosomes isolated from all these cell lines. And then they look at the wound closure. So as you can see from the pictures here, the gap creation is rather quite well defined and uniform. So if you look at the plotted graph, the error bars are relatively small. This shows that the results of data that they generate using the inserts are highly reproducible. So the take-home message for this experiment is that it showed that the exosomes released from the non-small cell lung cancers, they are able to enhance the angiogenic behavior of the endothelial cells. Another assay for the measuring of migration would be the Chemotaxis assay, which is the analysis of directed cell movement towards a chemoattractant. So one of the most common methods to perform chemotaxis assays is to use the Boyden chamber. So this is also known as a transfer assay. However, there are several limitations to the Boyden assay. In the Boyden chamber, as you can see from here, just look at the diagram, cells migrate from one side to the other side of a filter or a porous membrane. So in this case, single cell analysis cannot be tracked as migrating cells are counted at the opposite end of the membrane here, and therefore migration cell path cannot be analyzed. It also has a steep gradient and inhomogeneous cell distribution at the experimental starting point. To overcome these limitations, we can make use of the Ibidi microslide chemotaxis for live cell and high-resolution imaging. This slide is suitable for 2D and 3D assays for up to 48 hours. It allows for the creation of a precisely defined, stable chemotactic gradient. The cells are distributed homogeneously at the experimental starting point, and the slide also allows for single-cell analysis. So as you can see over here, this is the slide. It has 3 separate chambers for parallel chemotaxis assays. So over here is a cross-section. So what you can see is that you see the cells in here, which is the observation area. So this is where you will view your cells, and these are the 2 chambers where you can view it with media or chemoattractant. So let's look at a little video. So this is a video of HT1080 cells in a collagen 1 3D gel for 24 hours using the 10% FCS as a chemoattractant. So you can also see here that the individual cells can be tracked and their trajectory can also be plotted. Using our microslide chemotaxis solution, here is one application example published in Cancer Research early this year. In this experiment, lymph kinases 1-2, they were knocked down in the brain tumor glioblastoma cell line and then subjected to the chemotaxis assays using EGF as the chemoattractant. So they use EGF here. This is a lymph kinase 1-2 knockout cells. This would be the control cell. So as you can see here from the figures, the single cells can be tracked, and in the control cells, the cells are able to respond to the EGF signal and in a direction. There's a directionality can be observed. While down in the knockdown cells, they do not respond to EGF and they lost their directionality. So using this slide, you can generate such nice data. You are able to track single cell and observe their directionality. So let's move on to angiogenesis assays, which is one of the widely used assays to study the expansion of the vascular network, an important factor for tumorigenesis. Okay, so we have this microslide that is suitable for angiogenesis assay. It has a specialized geometry so that it is easy to conduct angiogenesis assays. Due to the well in a well, over here, as seen here, what do we mean by well in a well? Is just a little well here, in another well, yeah. Due to the well in a well format, there is no meniscus formation with all the cells in one focal plane. The slide is suitable with a broad range of gels such as matrigel, collagen, and agarose. Only 10 microliter of gel is needed to fill this inner well of the slide, making it very cost-effective. As many of you here would know that matrigel can be quite costly at times. So once the gel polymerized, the cells can then be seeded over here or into the upper well, incubate and ready for observation. Okay, so this is a time-lapse video. Let me play it. Okay, of the cuvette cells on matrigel for tube formation assay. So as you can see here, over a period of 10 hours, we can see really nice tube formation. Okay, this slide is also ideal for sprouting assays. So to carry out a sprouting assay, spheroids or pieces of tissues, they can be placed onto or directly in a gel before imaging. So I'll show you a little video to show how this is one cuvette cells embedded into a 3D gel made of collagen type 1. So you can see that the sprouts are forming from the spheroid. So with the fictive wells, the microslide angiogenesis is designed for low throughput assays. For large-scale experiments applications, we also have a 96-well plate format. In this application example, the investigators used microslide angiogenesis to investigate the effect of gene silencing or overexpression on tube formation. Mitochondrial lawn was first overexpressed or knocked down in the human oral squamous carcinoma cancer cell line, or CEM. The conditioned media were then collected and then incubated on the endothelial cells. So there are 2 endothelial cells used in this case, the cuvette and 3B11. So the conditioned media, they are collected and incubated on these cells to test whether they induce the angiogenesis of the endothelial cells. So what we found was that the conditioned media from the non-overexpressing cancer cells induces the tube formation of the endothelial cells, while that of the knockdown inhibits the tube formation. So you can see this from the graphs here. So the take-home message from this experiment is that the mitochondrial lawn induces production of inflammatory cytokines, which consequently activates angiogenesis. So this is one example of how you can investigate the effect of gene manipulation on tube formation. Similarly, this assay can also be used to screen compounds and drugs that may affect angiogenesis. So liquids are a crucial component of every living species. Many cell types are surrounded by moving fluids. Therefore, cell culture under flow would better stimulate the mechanical stimulus and induces a more physiological in vivo-like behavior. Intravasation is one of several carcinogenic events that initiate the escape of cancerous cells from their primary sites. So, cancer cells with a high metastatic potential are able to pass the endothelial cell barrier, which is this thing. You can pass it through after reaching a blood or lymphatic vessel. This trans-endothelial migration allows them to enter the bloodstream and then be transported to distant locations in the body. But once in the blood flow, the cancer cells are exposed to new environmental conditions like the shear stress. So once circulating in the blood flow, the cancer cells may eventually change and they can then adhere to the endothelial cell wall here. This rolling and adhesion of cancer cells is important in order to withstand the shear stress and be able to pass the endothelial cells again. So after another round of trans-endothelial migration, the secondary tumor begins at the new site. We are able to provide a whole package solution for cell flow assays. We have channel slides that comes in different channel heights and geometries for different experimental conditions. We have pumps and software for the analysis. So what you do is that cells are first seeded in the channel slide here. So after which the cells, the channel slide is then connected to the Ibidi pump system, and then flow can then be applied. So under the flow, right, the cell culture media is continuously pumped through the channel slides. So show you a little video of how the cells would look like under flow. So under the flow, so this results in a shear stress that influences the cell behavior and physiology. So the flow type, the flow rate, and the assay duration would depend on your experimental setup. The cells can then be imaged using live cell microscopy here. Images or videos of the flow conditioned cell layer can be directly acquired within the channel slides. So the cells are all seeded here. So the cells can also be harvested for the endpoint experiments such as like PCR or Western blot analysis. Here is one application example of using the microslide lure for the flow experiment. So in this experiment, the bone marrow endothelial cells, they were cultured in the channel slide. They were seeded on this channel slide. Then they were then treated with either isoproterenol or PBS, which is acted as a control. After which, GFP positive breast cancer cells, which is the MDA MB231, these are all GFP positive cancer cells. They are then added to the endothelial cells as flow. So the cells are keep flowing through this seeded endothelial cells, which is already seeded here. So after 3.5 hours, the wells were then washed in PBS and the relative adhesion of breast cancer cells to the endothelial monolayer was quantified by fluorescence labeled cell count assay. So if you look at the picture here, the blue is standing for the nucleus, the green is the GFP positive breast cancer cell. So as you can see, it is quite obvious that in the isoproterenol treated endothelial cells, right, you see more attachment of the GFP positive cancer cells. So this can be seen in this plotted graph as well. So in this experiment, the take-home message is that the isoproterenol stimulates the bone marrow endothelial cells to release certain factors that favor the adhesions of the breast cancer cells to the endothelial cells. So this is one example of assay that one can use to look at the effects of cancer cell adhesion. Moving on, we now know that there is a growing interest in using 3D cell culture for cell-based assays, as the majority of cells in the living tissue grow in a three-dimensional microenvironment in the ECM, the extracellular matrix. 3D environment also better reflects the in vivo situation more accurately. The 3D culture also allows co-culturing of two or more different cell types and is a better model for predicting in vivo responses to drug treatment. So when cultured in a traditional two-dimensional in vitro environment, the cells are attached to a flat surface and can grow and migrate on the substrate. So they can only grow and migrate on this 2D substrate here. In a 3D in vitro setup, the cells are either grown in suspension on a non-adhesive surface, or they can be embedded in or on a 3D matrix shown here that mimic the ECM and allows them to grow in all three different in the three directions. So one widely used 3D culturing technique is the application of multicellular spheroids. Spheroids are microscale spherical cell clusters which grow free of foreign material. So there are cells that adhere to each other under 3D non-adherent culture conditions. Spheroids are a useful model for examining tumor cell behavior. We have different solutions for supporting 3D culture. Micro-dish 25mm high bioinert allows long-term culture and high-resolution microscopy of spheroids on a non-adherent surface. As mentioned earlier, bioinert is completely non-adherent and allows no binding of any biomolecule, any proteins, even in long-term experiments. The bioinert surface is ready to use. There is no pre-hydration step, so it is suitable for high-resolution microscopy without any destructive autofluorescence. The microslide and microplate angiogenesis, which I mentioned earlier, can also be used for 3D culture. So the gel layer is directly connected to the medium reservoir above, which allows for fast and easy medium exchange by diffusion. I have also demonstrated earlier that the use of the microslide chemotaxis for cell migration. Although the slide can be used for 2D culture, culturing of cells in a 2D environment during a chemotaxis assay usually does not reflect the in vivo situation. Therefore, in order to mimic the natural environment, it is recommended to embed the cells in a 3D matrix to perform the chemotaxis assays. If you want to couple 3D culture and perfusion experiments, we have a solution here. We have a microsite 3D perfusion to allow spheroids or organoids cultivation in or on a gel layer or embedded in a 3D matrix. So this slide allows for superfusion with a low flow rate. This setup makes long-term cultivation possible for up to several weeks with an optimal nutrient supply. I would like to share with you a developing technology called the micropatterning. This technology enables spatially defined cell adhesion for spheroid generation, long-term culture, and high-resolution imaging. So defined adhesion spots are able to catch all the adherent single cells from a cell suspension. The surrounding bioinert is fully non-cell attachable, so this forces all the cells to aggregate to each other at the adhesion spots here, thus forming spheroids in a defined and controllable way. So using this setup, cells in 3D can be studied during proliferation, differentiation, invasion, and migration. So this technology is currently under development, but if you are interested in this technology and would like to customize your own pattern for your specific application, please send a request to us. Okay. We also have a full range of other complementing products from Biotium. Biotium is a leading life science supplier for high quality and innovative fluorescent tools that are able to assist with your cancer research. So we have kits for apoptosis assays, such as we have the adenosine V conjugates for the staining of phosphatidylserine at the cell membrane. We have this new field of caspase 3 substrates for detection of caspase activity. We have tunnel kit CF dye conjugated tunnel kits for the detection of DNA fragmentation. And we also have the CF dye conjugated mitobutyl dyes for the measuring of the change in mitochondrial potential. There is also a whole range of kits for viability and proliferation assays, which are either absorbance, fluorescence, or luminescence based. So you can contact us for more information on these kits. For immuno-fluorescence assays, we also offer a variety of stains for different organelles and cellular structures. The new spot and red dot dyes for the staining of nucleus. New spot live stain is specific, non-toxic, and does not require a wash step, and staining can be continuously monitored for several days. The new spot live 650 is particularly suitable for super resolution imaging techniques if anyone here does super resolution imaging. Calcein-AM and the Immuno-Fluorescence cell proliferation kits can be used for cytoplasm labeling. The cell bright and membrite are used for cell surface staining. These dyes can tolerate permeabilization and metamorphosation, allowing membrane staining to be combined with intracellular staining. So we also have a cytoskeleton probes to stain the mitochondria and actin using phalloidin. So as mentioned earlier, we also have microview dyes for staining the mitochondria. So these dyes stain mitochondria rapidly without a wash step and are non-toxic to cells for live cell imaging. There are also options for mitochondrial potential dependent or independent staining. Intracellular lipid droplets can also be stained using the lipid spot dyes. We also have lysoview dyes to stain the lysosomes. Staining is highly specific. There is no wash staining of the acidic organelles. We also have conjugates for vesicle trafficking, such as the CF dye dextran conjugates, transferrin conjugates, cholera, toxin subunit B, and many others. So now I would like to share with you to introduce you to the CF dyes, which is developed in-house by the biochems industry leading chemists. The CF dyes are a series of highly water-soluble fluorescence dyes that span the visible to the near-infrared spectrum. So these CF dyes, they offer superior brightness, photostability, and better signal-to-noise ratio when compared to other commercially available fluorescent dyes, such as the most commonly known ones, the elixir. So there are over 30 CF dyes across the spectrum. The near-IR dyes are ideal for near-IR Western, such as, usually for those people who use the Leica Odyssey, and also for small animal imaging. So certain CF dyes are also specially designed for super resolution microscopy. And due to their excellent labeling efficiency, CF dyes are ideal for labeling of the biomolecules, such as the proteins and nucleic acids, and we have kits available for labeling them. We have a range of CF dyes products that are already conjugated. So these include like bioconjugates such as the phalloidin, annexin V mentioned earlier, labeled nucleotides, anti-tag antibodies, primary antibodies, and secondary antibodies. For immuno-oncology research, we also have a panel of mouse and human reactive antibodies for flow cytometry. Tombow Bioscience products are comparable to other competitors. Apart from flow antibodies, it also offers a range of recombinant proteins for cell culture, proliferation, migration, differentiation, and other studies. We offer an expanded comprehensive suite of human and mouse chemokines, cytokines, growth factors, and other proteins. All recombinants are lyophilized and formulated as carrier-free for flexibility in the in vitro or in vivo bioassays. We also have a range of uncoated ELISA kits that are available in 2 or 10 well sizes and provide users with a budget-friendly solution compared to pre-coated immunoassays. So if you would like to find out more, you may write to us for more information. I'd like to thank you for your time, and I hope this webinar is useful to you and offers solutions and ideas for your experiments. So if you have any questions, you can type in your question in the chat box, or you may also write to me.